Buenos días a todos. Es para mí un honor.
Good evening. It's an honor for me to be part of this conference, this 7th International Scientific Digital Symposium. Well, in this case, it's digital. And I am very happy to be here with you today and to talk about the early programming of the immune system is a challenge. And we already heard in the previous session how Dr. Collada and Dr. Ryan have spoken about this topic, and we're going to insist on this topic because we believe that it's very interesting today, because the immune system starts developing throughout gestation and pregnancy with an adaptive immunity. You see on this chart how it starts in the first term of pregnancy with some dependent T cells and B cell dependent. And in the second and third term of pregnancy, we start seeing a release of some interleukins that are associated with innate immunity. But until birth, what we mainly see is a transfer of the IgG maternal antibodies, and the child will also receive the IgA antibodies through breast milk, and the IgG antibodies that go through the placenta to the fetus in the last part of gestation have a very important role in the tolerant response to food and other allergens, and with regard to the immune response of the infant. Additionally, after birth, we see the T cells and the natural killer T cells and the T regulatory cells, and we see higher levels of interleukin 10 and 12 and also interferon alpha. And in the case of interleukin 12, we can see how it starts at a low level and then it later increases. However, the development and maturity of the immune system doesn't end until adulthood in many cases. And it's important to bear in mind that we have this early programming and that there are some factors that are very important for the development of the immune system. And we also know that the development of the immune system is very much associated with some chronic diseases with that are inflammatory autoimmune diseases. So there are different factors. For example, maternal malnutrition, obesity, or diabetes, or the activation of the maternal HPA axis, or an impairment of the maternal immunocompetence, and there is a lower maternal IgG placental transfer, or a reduction of human immunological factors. All this can have an effect, an impact on the fetal development and on the development of the immune cells that are learning, so to speak, to provide certain responses after birth. All this is associated with the development of the infant at a metabolic level, and this is associated also with chronic inflammatory diseases such as the metabolic syndrome, cardiovascular diseases, cancer, or diabetes, or autoimmune diseases. And today we know that there are also epigenetic factors that will have an impact on the development of immune cells. If we take into account all these factors that are involved in the development of the immune system and the development of allergies, we can see the fact that they are factors that have to do with the microbiota, such as the type of delivery, the genetic predisposition, the use of antibiotics during pregnancy, unhealthy lifestyle, environmental contaminants, the type of diet, the status of the maternal microbiota and how it's transferred to the fetus, and the fact of receiving feeding breast milk or formula. So we have these epigenetic factors that can have an impact on the programming of the immune cells, and this will lead during gestation to a transient hematopoiesis of these cells, and this will determine how they are going to behave and their function in the case of macrophages and mast cells. And all this will impact, depending on this imprinting, will be determining the presence of long-term diseases. This slide is particularly interesting because it assesses how nutritional parameters are very important during the first stages of life. We see how maternal nutrition plays a very important role in the increase of risk of having diseases that are associated with the immune system or chronic diseases that are inflammatory. And depending on the mother's diet, if we have a mother that takes an appropriate number of calories, then there'll be a better fetal development. And if the mother also takes N3 PUFAs, this will reduce the risk of atopia. And if the mother has got dysbiosis, this will have an effect on the immunity. It will lower the antibodies that are transferred to the offspring. And if we have maternal malnutrition, there'll be a reduction in the child of the immune response to vaccines. And if we have malnutrition in mothers with zinc deficit, then there's an increase of cortisol levels, and this can have an impact on the patient's erythropoiesis. Additionally, when the child is born, it's important to see the difference between breastfeeding and formula, because breastfed infants will have an important intake of nutrients and will also receive bacteria, microbiome from the mother that will lead to an improvement of the innate immune system, and they will also be receiving oligosaccharides that will protect the gut mucosa that's under development, and also the immunoglobulins. And we shouldn't forget that the immune system continues to develop until 2 years of age, and therefore nutrition has a key role, and we reduce the risk of allergies and inflammatory diseases if there is a nutrition rich in fruit and vegetables. And children who have a deficit of iron, vitamin B12, vitamin D, and zinc will see a negative impact on their immune system. And also in children with a reduced protein intake, this can lead to an atrophy of primary lymphoid organs, and this can alter their immune development and increase the risk of diseases in the long term. Therefore, nutrition has a wide impact, and for example, in the case of allergies, we see that a high intake of vitamins, minerals through fruit and vegetables and will have an effect in the same way as the intake of prebiotics and probiotics or fiber that can be protective, and the same happens with N3 PUFAs. And therefore, we can say that it's possible to design a diet, or we can intervene with a diet to prevent diseases during this critical period from the prenatal phase until 2 years of age. So in this way, we can stop these diseases from occurring, from developing. And why is that? Because we have to take into account the timing of the GI tract that is very long in humans and doesn't end until childhood, until 6 or 7 years of age or even later. Therefore, the most sensitive stage would be the prenatal stage and the postnatal stage until 2 years of age, where there is a development of the epithelial cells and of the gut mucosa. Additionally, during this time, there's an implementation of the microbiota. It's the initial touch point with foodstuffs, of the body with foodstuffs, and the reactions in the GI tract will have an impact on the development of the immune system. I like to highlight the fact that the shaping of the immune system starts with the maternal microbiota, and during pregnancy and lactation, there is an imprinting of the gut microbiota that will determine the reaction of the child when faced with different stimuli, different allergens, and this will influence the infant's development depending on the child's nutrition, on genetic factors, the health of the mother, environmental factors, stress, infections, medication, and so on, and the type of diet and the use of prebiotics during these very critical stages of development. I have to say that when we are talking about the early programming of immune system, I have to say that breastfeeding provides the best nutrition to newborns, to infants, and compared with formula-fed infants, the growth patterns are different. We also see different nutritional status, a different gut microflora, and fewer infections in breastfed infants versus formula-fed infants. And there's also a different immune system development and immune response, and a better neurodevelopment, and there's an innate programming of the taste that the infants will prefer, and a prolonged exclusive breastfeeding will reduce the risk of GI respiratory infections and hospitalization for infections, even in developed countries. Therefore, breastfeeding has many important advantages. Firstly, we have the transference of multiple immune factors, and it promotes the gut immunity, and it also has an anti-inflammatory effect. Everything indicates, therefore, that breast milk is the perfect nutrition for the development of the immune system. In fact, breastfeeding transfers in a passive way IgA antibodies. It also regulates the development and the function of the gut permeability. Furthermore, it offers bioactive antimicrobial factors to the child, and it has a modulating and an immunoregulating effect on the evolution of the immune system, and it offers anti-inflammatory and antimicrobial agents that are very important during the early stages of life. And breastfeeding includes some very important ingredients such as proteins and lipids and carbohydrates and vitamins and minerals. And additionally, it offers bioactive components such as B and T lymphocytes, macrophages, immunoglobulins, growth factors, FGM components, the LC, PUFAs, the HMOs, probiotics, prebiotics, and other bioactive peptides that will have a very important impact on the development of the infant's immune system. And human breast milk also includes non-nutritive biomolecules such as bacteria, archaea, viruses, protozoa, anaerobic fungi, and lactic acid bacteria that are important and very well known due to their beneficial roles, such as Lactobacillus casei, Lactobacillus fermentum, and also the Bifidobacterium and the Bacteroides and the Clostridia class that will also be implanted in the gut and that are very important for this immune system development. We are talking about the core human breast milk bacterium that has lactobacillus, staphylococcus, streptococcus, and proteobacterium. All these bacteria of the bacterium may seem pathogenic, but when received through breast milk, then these bacteria will have a very important role in a positive development of the infant. And this microbial pattern in the gut, as I say, is very important. So breast milk has an important immune modulation effect that will sustain and support and regulate the cell population in the immune system and will also help with the maturation of the infant gut and associated lymphoid tissues, and also through the cytokines and the chemokines and the PUFAs, the MFGM, the nucleotides and glycoproteins and oligosaccharides. And all of this will be associated with the development of diseases such as allergy or asthma. So breast milk versus infant formula will create an immune tolerance network at the gut that is based on the interaction between the microbiome and the gut immune system, and this favors the optimal development of the infant's immune system. And I'd like to highlight the HMOs as very important components. Some mothers secrete HMOs, some others don't. And HMOs are in breast milk and they're associated with the microbiota of the mother. And this differs depending on whether the mother secretes HMOs or not. And the HMOs will drive the infant gut microbial colonization and the maternal secretor status will also have an impact on the neonatal gut microbiota. It's not the same to have a mother who secretes HMOs versus one that doesn't. And when there's a disruption of the early microbiota colonization, this has been associated with the risk of disease. Therefore, it's important to have this HMO secretion and to have an optimal microbiome in breast milk. Another important component that also impacts the development of the immune system is the MFGM. And there are several studies that confirm its effect on the development of the brain. And there's very important data regarding the benefits for the immune system development and the gut. And we see a lower prevalence of diarrhea in infants that are receiving formula supplemented with these components, and there's a reduction of otitis media, and there's also a bactericidal activity against several foodborne pathogens and a lower incidence of gastrointestinal and respiratory infections. Additionally, we see a benefit on the development of the intestinal epithelium. There's a protection against inflammation induced by Clostridium difficile toxin, and we observe a better microbiota profile that's more similar to that of subjects receiving breast milk, and it also has a moderate effect on the shaping of the gut microbial activity and function. When the MFGM and probiotics are added to the nutrition, this will have a significant impact on the composition of the microbiota. Lastly, I would like to make a reference to complementary supplementary nutrition and its impact on allergies, because it's maybe possible to have some primary prevention with an early exposure to food allergens to induce tolerance and reduce allergies. And it has been shown that the timing of allergenic food introduction is not associated with other allergic outcomes. And although the findings cannot be generalized beyond food allergy, we see that there is this mediation when there's IgE antibodies. And I wanted to mention this slide because the immune system continues to develop, and it's important to mention the importance of supplementary nutrition. And today, we shouldn't delay this supplementary nutrition to avoid the development of allergies. I would now like to present some very interesting findings from a study, the Cognis study, where we assess two different formulas. One is an experimental formula that was supplemented with MFGM and symbiotics. And here we are going to see the effect on the development of the immune system and on infections. So this is the Cognis study. This is the design. We had 85 children on the standard formula, 85 on the experimental formula, and 50 with that were breastfed, and we look at the development of the immune system, the evolution of the fatty acids and their concentration in the gut mucosa, and we saw changes in the percentages of arachidonic acid until 18 months, and we see the differences between the standard formula and the experimental formula. And we see differences in the gut microbiota among the different groups. For example, in the experimental formula group at 6 months, we see a greater presence of lactobacillus that's more similar to that of infants that were breastfed. And the same happens with at 12 months, and at 18 months, we also see a higher level of bifidobacterium. Therefore, during the first year, the gut microbiota of the children fed with the experimental formula was more similar to the microbiota of the breastfed infants and different from those fed with the standard formula. Here we see the maturity of this gut microbiota. With breast milk, there is a slower maturation process versus the formulas, and at 18 months, this variability is not so obvious. But the experimental formula always shows to be much more similar to breast milk. This is the comparison of the two formulas versus breast milk. In green, we have the experimental formula that is more similar to breast milk at 6 months and at 12 months and at 18 months. And the functionality is more similar to that of breastfed infants, and it's different from the functionality of infants that receive the standard formula. Here in these charts, we can see many differences in the metabolic activity of the bacteria when we compare the standard formula versus breastfeeding, and then we see that the experimental formula shows a very similar effect to that of breast milk. And in terms of infections, there is a lower incidence of infections at 12 months, gut infections and respiratory infections, and fewer number of visits to the pediatrician, and fewer events of fever and viral infection, and fewer consultations to the pediatrician in those receiving the experimental formula versus the standard formula. And when we associate the gut bacteria with these events, we see that at 6 months there is bacteroiditis that are associated more with vomiting and diarrhea. The same happens with proteobacteria at 18 months. It's also associated with vomiting, and the higher prevalence of firmicutes protects from vomiting and diarrhea. Therefore, firmicutes seem to be less beneficial when looking at obesity. However, we see the results here with the vomiting and diarrhea. And when we look at the three groups, breast milk has a higher presence of firmicutes in the gut. The standard formula mainly shows bacteroiditis, and in the case of the experimental formula, it also promotes in the second place the growth of a gut firmicutes. So it seems, however, here firmicutes in the standard formula is very, very low. So this effect has to do with the similar function that we have seen between breast milk and the experimental formula. When we look at the specific relationship with the secretion of salivary IgA, there is a negative association with the secretion in the case of bacteroiditis. However, firmicutes do promote a higher level of salivary immunoglobulin. Therefore, by way of conclusion, we can say that the immune system development depends on healthy establishment and maturation of the gut microbiota. Additionally, early nutrition has a significant impact and determinant effect on gut microbiota establishment, maturation, and function. The experimental formula that has been enriched with MFGM and LC-PUFAS and symbiotics promotes a gut microbiota that's more similar to that observed in breastfed infants, both structurally and functionally, and the immune system development in this case has high concentrations of salivary SIgA that seems to be associated with a higher amount of firmicutes in the gut that will protect from infections, and there's a reduced gap between formula-fed and breastfed infants, although we need more trials to confirm this data. Thank you very much for your attention. I hope you continue enjoying this event. Thank you.